Here we describe the process of RNA synthesis by RNA polymerase. RNA synthesis creates four types of RNA molecules: messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA, rRNA, and special function RNA. Most RNA are synthesized using DNA as template. However, there are viruses that use RNA as a template. This is a 3-lecture series that will cover 1. DNA-dependent synthesis of RNA, 2. RNA processing, and 3. RNA-dependent synthesis of RNA and DNA. Ribonucleic acids play 3 well-understood roles in living cells. Messenger RNAs encode the amino acid sequences of all the polypeptides found in the cell. Transfer RNAs match the anticodon to the messenger RNA while carrying a specific amino acid used for protein synthesis. Ribosomal RNAs are constituents of the large and small ribosomal subunits. Ribonucleic acids play several less understood functions in eukaryotic cells. MicroRNAs regulate the expression of genes, possibly via binding to specific nucleotide sequences, and ribozymes are catalytic RNA molecules that act as enzymes. Ribonucleic acid acts as genomic material in viruses. RNA is transcribed by RNA polymerase. Transcription is tightly regulated in order to control the concentration of each protein. mRNA are modified via splicing events to remove introns and join exons, polyadenylation at the 3' end, and capping at the 5' end. Ribozymes are also transcribed. These are SS or single-stranded RNA that fold into compact structures with specific catalytic functions. Ribozymes often use metal ions as cofactors. Here we are looking in the active site of RNA polymerase from E. coli. The RNA strand is synthesized complementary to one of the 2 DNA strands in a double helix. The DNA is transiently unwound. This causes positive supercoiling at the front of the transcription bubble and negative supercoiling behind it. The mechanism of RNA polymerization is very similar to that of DNA polymerization. The reaction involves 2 magnesium ions coordinated to the phosphate groups of the incoming nucleoside triphosphates. These are NTPs rather than the DNTPs of DNA. There are also 3 aspartic acids which are highly conserved in the RNA polymerase of all species. The purpose of the aspartic acids is to coordinate the magnesium ions. One magnesium ion facilitates attack by the 3' hydroxyl group of the alpha phosphate of the NTP. The other magnesium ion facilitates displacement of the pyrophosphate, and both metal ions stabilize the pentacovalent transition state. RNA polymerase unwinds 17 base pairs of DNA, around 17 base pairs, and covers about 35 base pairs of DNA. To begin transcription, RNA polymerase binds to a sequence called the promoter. The growing end of new RNA temporarily base pairs with DNA template of around 8 base pairs and inserts 50 to 90 nucleotides per second. The DNA duplex unwinds, forming a transcription bubble of around 17 base pairs. In this image, the transcription bubble moves from left to right along the DNA. The DNA is unwound ahead and rewound behind as RNA is transcribed. As the DNA is rewound, the RNA-DNA hybrid is displaced and the RNA strand is extruded. Here we're looking at some of the important landmarks during RNA polymerization. The direction of transcription is from left to right. The DNA template is in blue and the RNA being synthesized is in red. The DNA is circular, meaning that the region to the right of the transcription bubble, or in front of the RNA polymerase, at some point connects to the region to the left of the transcription bubble. The RNA polymerase separates the 2 strands of DNA as it travels along the DNA. Because the DNA is helical, meaning that it rotates along the axis of the DNA, RNA polymerase must rotate along with the DNA in order for DNA to be read. However, due to the size of RNA polymerase and the spatial constraints placed on it, it is unable to rotate along with the DNA. This forces the DNA to rotate instead. However, the DNA is also large and under spatial constraints and unable to rotate either. Thus, the DNA becomes positive supercoiled, also called overwound DNA, in front of the transcription bubble, and negative supercoiled, also called underwound DNA, behind the transcription bubble. There are two topoisomerases that relieve this tension as the RNA polymerase synthesizes RNA and reads the DNA. The topoisomerases are not shown here. The RNA polymerase is in close contact with the DNA ahead of the transcription bubble, as well as the separated DNA strands and the RNA within and immediately behind the bubble. A channel in the protein funnels new NTPs to the polymerase active site. The polymerase footprint encompasses about 25 base pairs of DNA during elongation. We will discuss the details of transcriptions in slides to come. The DNA template strand serves as the template for RNA polymerase. The DNA coding strand is the non-template strand and has the same sequence as the RNA transcript, with the exception of thymine being present in DNA and uracil present in RNA. There are also regulatory sequences in both strands. Unlike DNA polymerase, RNA polymerase does not have 3' to 5' exonuclease activity or proofreading capability. Thus, it has a high error rate of 1 in every 10,000 to 100,000 nucleotides. RNA polymerase is large with many subunits. Here you're looking at the core of the E. coli RNA polymerase. The RNA polymerase of eukaryotes is more sophisticated. The RNA polymerase holoenzyme has 5 core subunits: 2 alpha, 1 beta, a beta prime, an omega, and a sigma subunit. RNA polymerase binds to the promoter region of DNA to initiate transcription. This is a surface representation of the thermophilus RNA polymerase. The coloring scheme between the rotating structure and the cartoon on the bottom right are not identical, but you should get the point. The 2 alpha subunits function in assembly and binding to upstream promoter elements, shown in the next slide. The beta subunit is the main catalytic subunit. The beta prime subunit is responsible for DNA binding. The sigma subunit directs enzyme to the promoter, and each class of RNA polymerase holoenzyme has a different sigma subunit. The omega subunit appears to protect the polymerase from denaturation. This is a ribbon diagram of the same protein using the same coloring scheme as in the surface representation. These are the types of figures I'm expecting you to make in your reports. These figures were made with Chimera X, but you'll be using Chimera. Feel free to use Chimera X if you like. Holoenzyme is a catalytically active enzyme that includes all the necessary subunits, prosthetic groups, and cofactors for activity. Here are some examples of E. coli promoters that are recognized by RNA polymerase holoenzyme that contains a sigma 70. Remember that different sigmas will recognize different promoters. Sequences of the non-template strand are shown, read in 5' to 3' direction, as is the convention for representations of this kind. The sequence numbering begins with the transcription start site designated as +1. The sequences differ from one promoter to the next, but comparisons of many promoters reveal similarities, particularly in the -10 and -35 regions. The UP element, not present in all E. coli promoters, is shown in the P1 promoter for the highly expressed gene rRNB. UP elements, generally occurring in the region between -40 and -60, strongly simulate transcription at the promoters that contain them. The UP element in the rRNB P1 promoter encompasses the region between -38 and -59. The consensus sequence for E. coli promoters recognized by the sigma 70 is shown second from the top. Spacer regions contain slightly variable numbers of the nucleotides, designated as N. Only the first nucleotide coding the RNA transcripted Transcript at position 1 is shown. There are two consensus sequences at -10, TAT, AAT, and -35, TTG, ACA, for sigma subunit binding. These are called TATA sequences or TATA box. An AT-rich upstream promoter element between -40 and -60 binds the alpha subunit. AT-rich sequences promote strand separation. These sequences govern efficacy of RNA polymerase binding and therefore affect gene expression level. Nucleotides before the first nucleotide of the RNA molecules are considered upstream and given negative values. Before we jump into describing how and where RNA polymerase binds to DNA to initiate transcription, let's discuss how this was initially determined experimentally. The problem is as follows. You have a protein complex that binds to DNA and starts making RNA. Some simple questions are: 1. How does it know where to bind to DNA? 2. How does it know where to denature DNA? 3. How does it know where to start making RNA? And 4. How does it know where to stop making DNA? This slide shows the strategy used to identify the location on DNA where any protein binds to. You start with purified components. 1. A large piece of DNA where you think the RNA polymerase or the protein binds to. 2. Radio-label the DNA with P32. This is radioactive P32. 3. Bind protein to DNA in one tube and put DNA in another tube. 4. Treat both samples with chemicals or enzymes able to cleave the DNA. 4. Run the reaction products on gel electrophoresis and visualize the bands with X-ray film or an imager plate. The P32 incorporated into the DNA in step 2 will show a band on X-ray film. At the very top, we have our radio-labeled DNA. In tube 1, we add RNA polymerase and the DNA. In tube 2, we only have DNA. To each tube, you then add enzymes that can digest DNA. These are called DNase. The DNase is added at a low enough concentration such that there is only a single cleavage event in the allowed time of the reaction. Note that the DNase can cut at multiple positions. You denature the samples to remove the bound RNA polymerase from the DNA and run a page, and you visualize the page. On the page, you will see a single band for each cleavage event. Your initial substrate is homogeneous, but your products will be of different lengths. At the very top will be the uncut DNA fragment, and at the bottom will be the smallest DNA fragment. The bands in the middle migrate according to their site, their digestion site. By comparing the left to the right lanes, one can identify where the RNA polymerase binds because RNA polymerase binding to DNA will protect the DNA from digestion by DNase. If we know the sequence of the DNA we are using and know where the DNase will cut, we can figure out where the RNA polymerase binds to. This is an experimental result showing where RNA polymerase binds to the LAC promoter. In this experiment, the 5' end of the non-template strand was radioactively labeled. Lane C is a control in which the labeled DNA was cleaved with a chemical reagent that produces a uniform banding pattern. This means that the reagent digests one nucleotide at a time. The lane on the far left, the minus lane, does not have the RNA polymerase in the tube. And the lane in the middle, the plus lane, does have RNA polymerase in the tube. The lack of specific bands in the plus lane tells us where RNA polymerase may be binding to the DNA. RNA polymerase uses its sigma factor to bind to the promoter. This RNA polymerase has a sigma 70. In the next slide, you'll see a table of additional sigma subunits and where they bind to. In the initiation of transcription, the DNA-bound sigma factor recruits RNA polymerase to the DNA to create a closed complex where the DNA is not unwound. An open complex then forms, and the region from around -10 to +2 unwind. RNA polymerase moves away from the promoter, and the sigma factor is replaced by the protein NUS-A. RNA polymerase. In the elongation of transcription, RNA polymerase binds to triphosphate nucleosides and generates the RNA transcript. NUS-G binds to both the RNA polymerase and ribosome, linking the two together, directly affecting the rate of transcription in prokaryotic cells. Step 1. Sigma 70 binds to the promoter and recruits the RNA polymerase. This creates the closed complex. Step 2. This complex creates a transcription bubble and the open complex is generated. Step 3. Transcription is initiated and RNA polymerase begins to read the template strand of DNA as it creates the complementary RNA segment. Step 4. The RNA fragment is elongated as the RNA polymerase moves away from the promoter. Step 5. The sigma factor comes off as the RNA polymerase moves away from the promoter, and NUS-A moves in its place on RNA polymerase. Step 6. RNA synthesis continues till a signal is detected by RNA polymerase. NUS-A dissociates from RNA polymerase, and RNA synthesis is terminated. The entire process can be repeated. A number of steps have been occluded from this process for simplification. The 7 sigma subunits of E. coli RNA polymerase. The affinity of each sigma subunit for its promoter, the number of subunits per cell, their occupancy on the RNA polymerase, and their function are shown. Transcription is an energy-intensive process, so it's logical to regulate gene production. Regulation is achieved in many ways. One way is to regulate the affinity of RNA polymerase for a promoter. The promoter sequence can be altered, activator proteins may be required, and repressor proteins may block necessary binding sites. Termination of transcription in E. coli. Here we discuss two types of termination in E. coli. 1. Row-independent, and 2. Row-dependent. RNA polymerase pauses at a variety of DNA sequences, some of which are terminators. One of two outcomes is then possible. The polymerase bypasses the site and continues on its way, or the complex undergoes a conformational change called isomerization. In the latter case, intramolecular pairing of complementary sequences in the newly formed RNA transcript may form a hairpin that disrupts the DNA-RNA hybrid, the interaction between RNA and polymerase, or both, resulting in isomerization. In row-independent terminations, the RNA polymerase encounters a terminator sequence composed of adenines in the template strand. The RNA synthesized has a polyuridine at the end. An AU hybrid region at the 3' end of the new transcript is relatively unstable, and the RNA dissociates from the complex completely. This leads to termination. The self-complementary regions in the transcript forms a 15 to 20 nucleotide hairpin before the 3' end. This is the usual outcome at terminators. At other pause sites, the complex may escape after the isomerization step to continue RNA synthesis. Row-dependent termination of transcription. The presence of a run site, a common CA-rich sequence, in the transcript recruits the row helicase. The row helicase binds to the site and migrates along the messenger RNA in the 5' to 3' direction and forces the messenger RNA to separate from the RNA polymerase. 4 RNA polymerases have been discovered to date for eukaryotes. These include RNA polymerase 1, which synthesizes pre-ribosomal RNA that are precursors for the 28S, 18S, and 5.8S rRNAs. RNA polymerase 2, the subject of this image, which is responsible for synthesis of messenger RNA. This polymerase is very fast incorporating 500 to 1,000 nucleotides per second and can recognize thousands of promoters. We'll talk about this in the next slide. Each polypeptide chain in this structure is colored differently. This holoenzyme is clearly more sophisticated than its prokaryotic counterpart. The other two RNA polymerases are RNA polymerase III, which makes transfer RNAs and small RNA products, and plants appear to have RNA polymerase IV that is responsible for the synthesis of small interfering RNAs. Mitochondria have their own polymerase. Features of some promoters recognized by eukaryotic RNA polymerase II, also called Pol2. The TATA box is the major assembly point for the proteins of the pre-initiation complexes of Pol2. The DNA is unwound at the initiator sequence INR, and transcription site is usually within or very near the sequence. In the INR consensus sequence shown here, N represents any nucleotide and Y represents a pyrimidine nucleotide. Many additional sequences serve as binding sites for a wide variety of proteins that affect the activity of Pol2. These sequences are important in regulating Pol2 promoters and differ greatly in type and number. And in general, the eukaryotic promoter is much more complex than suggested here. Many of the sequences are located within a few hundred base pairs of the TATA box on its 5' side. Others may be thousands of base pairs away. The sequence elements summarized here are more variable among the Pol2 promoters of eukaryotes than among the E. coli promoters. The majority of Pol2 promoters lack a TATA box or a consensus INR element, or both. Additional sequences around the TATA box and downstream to the right as drawn of INR may be recognized by one or more transcription factors. Eukaryotic mRNA transcription involves many proteins to generate a large macromolecular complex. This process involves many highly conserved transcription factors. RNA polymerase II is a well-studied system. It has 12 subunits, and some of the subunits have structural homology to bacterial RNA polymerases, suggesting that they share a common ancestor. Proteins required for initiation of transcription at the RNA polymerase II promoters of eukaryotes. RNA polymerase II is also referred as Pol2. RNA Pol2 has 12 subunits. The additional proteins involved in transcription, for example, TBP, TF2A, and TF2D, have additional subunits. Thus, the macromolecular complex necessary for initiating, elongating, and terminating transcription is quite complicated. The assembly of RNA polymerase at the promoter is initiated by the TATA binding protein, TBP, with the promoter. TBP is part of a multi-subunit complex, TF2D. Other proteins in this complex include TF2B, TF2A, TF2F, TF2E, and TF2H. The helicase activity of TF2H unwinds DNA at the promoter. The kinase activity of TF2H phosphorylates the polymerase at the carboxy terminal domain, changing the conformation and enabling RNA polymerase II to transcribe. After 60 to 70 nucleotides, TF2E is released, which is followed by TF2H release. Elongation factors bound to RNA polymerase II enhance processivity and coordinate post-translational modifications. Some elongation factors are bound to the phosphorylated CTD. For termination, Pol2 is dephosphorylated. The regulation of Pol2 is quite complex, and unfortunately, we don't have time to discuss this. Transcription using RNA polymerase II. The sequential assembly of TBP, often with TF2A, TF2B, TF2F plus Pol2, TF2E, and TF2H results in a closed complex. Within the complex, the DNA is unwound at the INR region by the helicase activity of TF2H and perhaps of TF2E. creating an open complex. The carboxyl terminal domain of the largest Pol II subunit is phosphorylated by TF2H, and the polymerase then escapes the promoter and begins transcription. Elongation is accompanied by the release of many transcription factors and is also enhanced by the elongation factors. After termination, Pol II is released, dephosphorylated, and recycled. Transcribed genes are more actively repaired than silent genes. This may be partly explained by the fact that TF2H also has a role in nucleotide excision repair, as it recruits the NER complex at a lesion. Genetic repair diseases are associated with TF2H defects. Maturation of mRNA in eukaryotes. The mRNAs synthesized in eukaryotes are further processed before translation. This is protein synthesis can occur. This includes the addition of a 5' cap, cleavage, polyadenylation, and splicing. The 5' cap, shown in red, is added before synthesis of the primary transcript is complete. A non-coding end sequence, an intron colored in orange, follows the last exon. Splicing can occur either before or after the cleavage and polyadenylation steps. All the processes shown here take place in the nucleus. Dozens of proteins coordinate with one another and with proteins involved in RNA transport to ribosomes to accomplish this task. An unprocessed, newly synthesized RNA molecule is referred to as the primary transcript. Again, processing includes splicing out introns and rejoining any exons for a continuous sequence, adding a 5' cap, adding a 3' polytail, and mRNA degradation. The 5' cap, 7-methylguanosine, M7G, is joined to the 5' end of almost all eukaryotic mRNA in an unusual 5' triphosphate linkage. This is formed with a G molecule of GTP. Methyl groups, shown in light red, are often found at the 2' position of the first and second nucleotides. RNA in yeast cells lack the 2' methyl groups. The 2' methyl groups on the second nucleotide is generally found only in RNAs from vertebrate cells. The 5' cap protects RNA from nucleases and forms a binding site for ribosomes. Splicing. Most genes in vertebrates and some in yeast, a few in bacteria have introns. Exons are usually less than 1,000 base pairs in length, and introns are 50 to 700,000 base pairs in length, with an average of 1,800 base pairs. There's also far more intron sequences in a gene than exon sequences. The human genome has more than 200,000 introns spread across 20,000 genes. There are 4 classes of introns. Group 1 and Group 2 introns are self-splicing. They require no additional proteins or ATP. They are in nuclear, mitochondrial, and chloroplast genomes. They differ mainly in the splicing mechanism. Some are found in bacteria. Spliceosomal introns are spliced by enormous complexes called spliceosomes. These are the most common introns. They frequent in protein coding regions of eukaryotic genomes. The last class of introns are tRNA introns. These are spliced by protein-based enzymes. The primary transcript is cleaved by endonucleases, and the exon ends are joined by ATP-dependent ligase. Here we are looking at the splicing of group 1 introns. The 3' hydroxyl of GTP, GDP, or GMP acts as a nucleophile to break the phosphodiester bond between a U and A. The GTP, GDP, GMP becomes covalently attached to the intron. The 3' hydroxyl of the U-ending exon then attacks the 5' end of the other piece of exon to rejoin the pieces. The modified intron leaves. Here is the first step in the two-step splicing of group 1 introns. In this example, the 3' OH of a guanosine molecule acts as a nucleophile, attacking the phosphodiester linkage between the U and A residues at an exon-intron junction of an mRNA molecule. Group 2 introns. The chemistry here is similar to that of group 1 intron splicing, except for the identity of a nucleophile in the first step and formation of a lariat-like intermediate, in which one branch is 2' 5' phosphodiester bond. The 2' OH of a specific adenosine in the intron acts as a nucleophile, attacking the 5' splice site to form a lariat structure. The 3' OH of the 5' exon acts as a nucleophile, completing the reaction to create a spliced RNA and a lariat. Spliceosomal introns. Spliceosome introns are removed via a large complex called the spliceosome, shown here as a surface model. The spliceosome is made up of SNRPs, which stand for small nuclear ribonuclear proteins. SNRP RNA is called snRNA for small nuclear RNA, which are 100 to 200 nucleotides long. There are 5 snRNAs known in eukaryotes: U1, U2, U4, U5, and U6. GT at the 5' end and UA at the 3' end usually mark sites of splicing. U1 and U2 SNRPs contain regions that are complementary to messenger RNA. U1 helps define the 5' splice site, and U2 binds near the 3' end of the intron. U6 binds to U2, and U4 binds to U6. Base pairing of U1 to the primary transcript sequence helps define the 5' splice site during spliceosome assembly. U2 is paired to the intron at a position encompassing the A residue, shaded light red, that becomes the nucleophile during the splicing reaction. Base pairing of U2 snRNA causes a bulge that displaces and helps to activate the adenylate, the 2' OH of which will form the lariat structure through a 2,5 phosphodiester bond. Next, U2, U4, U5, and U6 bind, bringing at least 50 proteins to create a spliceosome. This is the ribbon diagram of the spliceosome shown on the left. ATP is required for assembly but not cleavage of the primary transcript. Some parts of the spliceosome are attached to the CTD of RNA polymerase, indicating that there is coordination of splicing with transcription. Note that the splicing mechanism is similar to the group 2 introns. Addition of the poly A tail. Pol2 synthesizes RNA beyond the segment of the transcript containing the cleavage signal sequences, including the highly conserved upstream sequence AAUAA. This cleavage signal sequence is bound by an enzyme complex that includes an endonuclease, a polyadenylate polymerase, and several other multi-subunit proteins involved in sequence recognition, simulation of cleavage, and regulation of the length of the poly A tail, all of which are tethered to the CTD. In step 1, the endonuclease cleaves the RNA at a point 10 to 30 nucleotides 3' or downstream of the sequence AAUAAA. In step 2, the polyadenylate polymerase synthesizes a poly A tail 80 to 250 nucleotides long, beginning at the cleavage site. The poly A tail serves as a binding site on messenger RNA and protects it from degradation. The surface representation on the left-hand side is the cleavage and polyadenylation specificity factor, CPSF complex. This multi-subunit complex is responsible for defining the polyadenylation site, primary transcript cleavage, and the poly-A polymerase recruitment, and for coupling 3' end formation with transcriptional termination. Overview of eukaryotic messenger RNA processing. The albumin gene has introns A to G and exons 1 to 7 and L. L encodes a signal peptide sequence that targets the protein for export from the cell. About 3/4 of the RNA is removed during processing. Pol2 extends the primary transcript well beyond the cleavage and polyadenylation site, labeled as extra RNA, before terminating transcription. Alternative splicing. A single gene can yield different peptides depending on the RNA processing. Splicing is performed at the 5' and 3' sites. In this example, you can have two alternating splicing events. The different messenger RNAs will produce different polypeptide chains. More than 95% of human genes are alternatively spliced. Poly-A site choice. In some primary transcripts, the poly-A tail can be placed in one of multiple locations. Here you're looking at a poly-A tail being placed in one of two locations. This generates greater diversity and is used in generating the variable domains of IgG heavy chain. Remember that the variable domain is where the IgG binds to its epitope. Processing of transfer RNA and ribosome RNA. Here are some modified bases of rRNA and tRNA produced in post-transcriptional reactions. The standard symbols are shown in parentheses. Note that the usual ribose attachment point in pseudouridine. This is just a small sampling of the 96 modified nucleosides known to occur in different RNA species, with 81 different types known in tRNAs and 30 observed to date in rRNAs. The 90S pre-ribosome transcript matures into the 40S and 60S, which then assemble to form portion of the eukaryotic ribosome. During transcription, the 45S primary transcript is incorporated into a nucleolar 98 pre-ribosomal complex, in which rRNA processing and ribosome assembly are tightly coupled. This process starts in the nucleus and finishes in the cytoplasm. In step 1, the 45S precursor is methylated at more than 100 of its 14,000 nucleotides, either on the bases or on the 2' OH groups. These are the red tick marks. Some uridines are converted to pseudouridine. These are the blue sticks. We talked about what pseudouridines are on the prior slide. And a few other modifications occur. These are the green ticks, and these are modifications such as dihydrouridine. Step 2. A series of enzymatic cleavages of the 45S precursor produces the pre-40S and pre-60S. Additional cleavages and export of these two into the cytoplasm produces the mature 40S and 60S. The ribosomal subunits gradually take shape with the assembling of ribosomal proteins. The cleavage reactions and all of the modifications require small nucleolar RNAs, or SNO RNAs. SNO RNAs are found in protein complexes, SNO RNPs, in the nucleolus that are reminiscent of spliceosomes. The 5S rRNA is produced separately. MicroRNAs are short, non-coding RNAs of around 22 nucleotides. They bind to specific regions of mRNA to alter translation. They assist in cleaving the mRNAs or blocking the mRNA from translation. About 1,500 human genes encode miRNAs, and one or more affect the expression of most protein-coding genes. These are synthesized from larger precursors. The larger precursors are processed by endonucleases called DROSHA and DICER. The primary transcript of miRNA is larger RNA of variable length, called a pri-miRNA. Much of its processing is mediated by two endoribonucleases in the RNA3 family. These are Drosha and Dicer. First, in the nucleus, the pri-miRNA is reduced to a 70 to 80 nucleotide precursor miRNA called pre-miRNA by a protein complex including Drosha and DGCR8. The pre-miRNA is then exported to the cytoplasm in a complex with a protein called exportin 5 and the RAN GTPase. In the cytoplasm, RAN hydrolyzes the GTP, then exportin 5 protein and the pre-miRNA are released. The RAN GDP and exportin 5 proteins are transported back into the nucleus. The pre-miRNA is acted on by Dicer to produce the nearly mature miRNA paired with a short RNA complement. The complement is removed by an RNA helicase, and the mature miRNA is incorporated into protein complexes, such as the RNA-inducing silencing complex, called RISC, which then bind a target mRNA. If the complementarity between miRNA and its target is nearly perfect, the target mRNA is cleaved. If complementarity is only partial, the complex blocks translation of the target mRNA. Ribozymes are RNA molecules that catalytically cleave RNA. They cleave themselves or another RNA. Their 3D structure is integral to proper function, meaning that the active site is made of nucleotides that are distant to one another in the primary sequence. They follow Michaelis-Menten kinetics. Their general mechanism follows nucleophilic attack of a sugar OH on phosphates followed by phosphodiester bond hydrolysis. Some well-characterized ribozymes are hammerhead ribozyme, self-splicing intron 1 group, and RNase P. Certain virus-like elements, or virusoids, have small RNA genomes and usually require another virus to assist in their replication. Some virusoid RNAs include small segments that promote site-specific RNA cleavage reactions associated with replication. These segments are called hammerhead ribozymes because their secondary structures are shaped like the head of a hammer. Hammerhead ribozymes have been defined and studied separately from the much larger viral RNA. A shows the minimal sequence required for catalysis by the ribozyme. The boxed nucleotides are highly conserved and are required for catalytic function. The arrow indicates the site of self-cleavage. The hammerhead ribozyme is a metalloenzyme. Magnesium ions are required for activity in vivo. The phosphodiester bond at the site of self-cleavage is indicated by an arrow. B shows 2D and 3D structure of a group 1 RNA intron from tetrahymena. RNA-dependent synthesis of nucleic acids. So far, we've seen RNA synthesized using DNA as a template, and this is the norm. However, viruses do not fit the norm. Retroviruses have genomes of single-stranded RNA and use a viral-encoded enzyme called a reverse transcriptase. The enzyme is used in reverse transcription to generate DNA. This DNA can be incorporated into the genome of the host. Because retroviruses incorporate the genomes into the host, the incorporation process can disrupt important genes and cause cancer. Retrovirus infection of a mammalian cell and integration into host chromosomes. Viral particles entering the host cell carry viral reverse transcriptase and a cellular tRNA, already base paired to the viral RNA. The purple segments represent the long terminal repeats on the viral RNA. The tRNA facilitates immediate conversion of the viral RNA to double-stranded DNA by the action of reverse transcriptase. Once converted to double-stranded DNA, the DNA enters the nucleus and is integrated into the host genome. The integration is catalyzed by a virally encoded integrase. Integration of viral DNA into host DNA is mechanistically similar to the insertion of transposons in bacterial chromosomes. This was covered in the lecture for DNA metabolism. For example, a few base pairs of host DNA become duplicated at the site of integration, forming short repeats of 4 to 6 base pairs at each end of the inserted retroviral DNA. This is not shown in the image. On transcription and translation of the integrated viral DNA, new viruses are formed and released by cell lysis. This is in the right-hand side of the image. In the viruses, the viral RNA is enclosed by capsid proteins called GAG and outer envelope proteins called ENV. Additional viral proteins, reverse transcriptase, integrase, and a viral protease are needed for post-translational processing of viral genomes. These are packaged within the viral particle with the RNA. Retroviruses typically contain 3 genes plus a long terminal repeat. The group-associated antigen, or GAG for short, encodes a long polypeptide that is cleaved into 6 smaller proteins that make up the viral core. The polypeptide, or POL, encodes a protease that cleaves the long polypeptide, reverse transcriptase, and an integrase to insert DNA into the host genome, and a viral envelope protein called ENV, which is responsible for cell membrane fusion. The long terminal repeats, or LTR, facilitate integration of virus genome into host DNA. The long terminal repeats, LTRs, have sequences needed for the regulation and initiation of transcription. The sequence denoted psi is required for packaging of retroviral RNAs into mature viral particles. Transcription of the retroviral DNA produces a primary transcript encompassing the GAG, POL, and ENV genes. Translation produces a polyprotein, a single long polypeptide derived from the GAG and POL genes, which is cleaved into 6 distinct proteins. Splicing of the primary transcript yields an mRNA derived largely from the ENV gene, which is also translated into a polyprotein, then cleaved to generate viral envelope protein. The reverse transcriptase catalyzes 3 reactions. 1. RNA-dependent DNA synthesis, 2. RNA degradation, and 3. DNA-dependent DNA synthesis. These enzymes contain zinc like DNA polymerases. They use a primer of tRNA and they lack 3' to 5' proofreading like RNA polymerases. This lack of proofreading creates a tremendous amount of mutations to occur when the transcriptases are working, approximately 1 in every 20,000 nucleotides. It is this error-prone activity that makes HIV such a successful virus. It allows the virus to experience countless mutations that allow it to escape from drugs and antibodies. Retrotransposons. Retrotransposons in eukaryotes have similarities to retroviruses. Retrotransposons are mobile genetic elements in eukaryotes. They encode an enzyme with homology to reverse transcriptase of retroviruses. They move between positions via RNA intermediates using their enzyme to make DNA from RNA, and are unlike bacterial transposons that move directly from DNA to DNA. Here we're looking at 2 retrotransposons. Most lack the on the gene so they don't form viral particles. However, there are genes for ENV-like proteins in eukaryotes. These proteins are indeed responsible for membrane fusion, and some of them have structural homology to the ENV proteins from viruses. This begs the questions: did these ENVs originate from viruses, or did these genes give rise to viruses? Telomeres are structures at the ends of linear eukaryotic chromosomes. They have tandem repeats usually rich in Tg, with AC on the opposing strand. They can be tens of thousands of base pairs long in mammals. The Tg strand is longer than its complement, leaving a 3' overhang of several hundred bases. The telomeres are not easily replicated using DNA polymerase. Beyond an end, there is no template for an RNA primer. This causes chromosomes to be shortened within each strand. Telomerase adds telomeric sequences to solve this problem. The internal template RNA of telomeres binds to and base pairs with the Tg primer of DNA. Telomerase adds more T and G residues to the Tg primer, then repositions the internal template RNA to allow the addition of more T and G residues that generate the Tg strand of the telomere. The complementary strand is synthesized by cellular DNA polymerases after priming for an RNA primase. Telomerase has RNA with Ca repeat to serve as template for synthesis of the Tg strand of the telomere. Telomerase binds to the 3' end of the chromosome and hangs off so that the RNA template extends beyond it. The gap on the bottom strand is filled by DNA polymerases.